All right, chapter 12.3 is going to deal with replication. So now we should know the structure about DNA, and we're going to move on to how it's replicated. So remember that we talked about replication of DNA before. We talked about the replication of DNA in mitosis. And if you remember back and look in your notes, it occurs during the S phase or the synthesis phase. I did talk to you about that. I said, hey, this is just where DNA copies itself. And I really didn't go really further deeper into the material there. I just said, hey, we're going to get to this when we get to DNA structure. So we're going to come back and kind of talk about the DNA structure and how it relates to the replication. Now, before that, again, this is one of those scientists that I don't really focus on. Messelson and Stahl. These guys were in 1958. They're trying to figure out how DNA replicates. So at this point, we've kind of figured out the structure of DNA. It was around this time that Rosalind Franklin took her the picture. I think it was 51 or 52 where she took the picture. Watson and Crick had built the first model. They wouldn't get awarded the Nobel Prize until 62, but we're starting to know about the structure. And we know that there's two strands and they have this helical form. Now, we had three different guesses. We had the conservative model, we had the semi-conservative model, and we have the dispersive model. And you can see each of these and how they are different. The conservative, you have both strands being a template for the new DNA. In the semi-conservative, each strand will be a template. You can see the old is blue and the new is yellow. And you can see the dispersive where you have parts that are new and old in both of the DNA strands. Now, in the second replication, you can see again, we're going to have one of these old strands and three new. We're going to have two new and then two that are kind of half and half. And in this one, we get like a kind of in the middle of each where we're going to have a mix of new and old. Now, there's only one that's going to win out, but these were our three guesses. And they did their experiment. And what they did was they tagged it with different nitrogen isotopes. And you can see the conservative versus semi-conservative and dispersive here, what they would hypothesize would happen. The actual results from the first one were that they had a mix of N14 and 15. So N15, it actually in the centrifuge, it disperses a little bit differently and it goes lower in the test tube. N14 goes a little bit higher. And the N14-15 will be in the middle of those two, and same with the N14-15 dispersive. So after the first round of replication, we kind of weed out the conservative model. It's not that model. If it was that model, we would see the two bands here, and we don't see that. In the second replication, the second round, again, we're still going to see that N14-15, but we're also going to see the N14 model and replicated DNA. And again, we don't see the only N14-15. We see them both here. So this proves that the semi-conservative nature of DNA replication is what we are looking for. And they do this time and time again. You can see this first generation, the second generation here. You're getting less and less of that N14, N15, and you're going to get more and more of the N14-14. So basically what this is saying is that DNA replicates semi-conservatively. And I know you're probably thinking like, wow, Mr. Fradov, like I never knew DNA replicates semi-conservatively, like where one side is the template for the other. Yeah, again, this is pretty easy for us to look back and say this is how DNA replicates. But back then, this was like huge news. They didn't know anything about DNA. They're just starting to figure out the structure, and then this comes along. It's like, oh my gosh, now we know how DNA replicates, and this really progressed the field very quickly. So just remember, like I said, DNA replication occurs during the S phase of mitosis. It has that semi-conservative replication nature. And again, that means one side of each of the double-stranded DNAs is going to be used as a template for the new DNA. So there are a bunch of different enzymes that we have to learn about in this chapter. Again, I think it's really cool. This is like one of the chapters that really shows you the difference between AP and regular bio. In AP, you're going to learn a lot more enzymes. In like regular bio, we only learn one or two enzymes, which is great. Again, you're just getting a taste of what this is. But in AP, you have to learn a lot more. So here we have topoisomerase. This is the first enzyme that we're going to learn about. I will forever remember this as Topio Tasty Cakes. My first year teaching this, I had a student. Shout out to that one student. I hope you watch this for some reason, but they called it Topio Tasty Cakes. And they literally on their test wrote it. I forget if I marked the point off for that or not, but it's the enzyme that just helps kind of relax the DNA and kind of smooths it out and makes the double helix, like the turns, go away. Helicase is the next enzyme. It's going to unzip the parent strand, so just the original strand of DNA there, and it's going to start breaking those hydrogen bonds. Remember, we said those hydrogen bonds are pretty weak within the bases. Here's just an image, a cartoon. Now just relax your backbone. This is topoisomerase, and you can see helicase breaking apart the DNA. They're actually just breaking apart the hydrogen bonds. All right, replication starts at the origin of replication. We're going to talk about that later on and what that means. Oh wait, no, I think the next slide actually does that. I have a pretty good analogy for that. And the replication fork. This is actually the location where helicase is breaking down the bonds. It's important to understand the difference between those two. So the origin of replication. If we go back here, the origin of replication. That's where it starts. This is the origin of replication. As you read, though, that's going to be the replication fork. So I'm reading this. This story begins on a beautiful day. So this is the origin of replication, and this is the replication fork. So we start somewhere, and then we continue on, and that's going to be the replication fork. I think that's a great analogy between the two. All right, so primase is going to be an enzyme that's going to come and it's going to attach itself to the DNA and it's going to attach RNA nucleotides to certain locations. These RNA nucleotides are going to be called primers, and these primers are going to be what DNA polymerase binds to. And starts when the polymerase binds to these primers, it's going to start bonding free floating nucleotides to the complementary bases, so like A's and T's, G's and C's. So these primers are pretty much just flags on the DNA that say, "Hey, DNA polymerase, come bond here. We're going to, this is where we want you to start." And then that DNA polymerase does that. It bonds and it starts bonding complementary strands of DNA. So you can see here, the DNA polymerase is going to start bonding these free-floating nucleotides found within a nucleus, and it's going to bond them A's, T's, G's, and C's. Apples in the trees, cars in the garage. So DNA polymerase is going to be an enzyme that works in a 5 to 3 prime direction, and most, if not all, enzymes. I'm going to go back. Work in this 5 to 3 prime direction. Again, I really can't name an enzyme that works in a 3 to 5 prime. I'm sure they're out there, but all the enzymes that we're going to talk about are going to work in a 5 to 3 prime direction, which means they make in a 5 to 3 prime. They don't follow, they actually follow in the opposite direction because remember, DNA is anti-parallel. They're going to go in opposite directions but parallel. So again, it makes in a, you can see here, the 5 to 3 prime direction. So this is what creates that identical strand of DNA during replication. This is on the leading and lagging strand. Now, because DNA is in a anti-parallel direction, there will be something called the leading versus lagging strand. The DNA polymerase will go in opposite directions and create something called Okazaki fragments. So we can see here the difference between the leading and lagging strand. Leading strand, the polymerase is going to follow the replication fork. In the other location or the other strand, it's going to go away from the replication fork. And as this unzips, as the helicase unzips, it's going to make the DNA in the opposite direction on the lagging strand. It has to do this because remember, the original DNA strand is in that opposite direction. So it has to move this direction this way and then towards the replication fork this way. Now, these little segments, because as this unwinds here and unravels the DNA, you're going to get short segments. These are called Okazaki fragments. And I remember learning this back in the freshman year of college and how they said, oh, this was just, where do you think this name came from? And I actually never knew. I was like, I don't know where it came from. It's what they named it. This is named after the scientist. He was a Japanese scientist that figured out the Okazaki fragments, and they named it after him. Let's see what else do we have here. Oh, okay. So after the Okazaki fragments are created, we have an enzyme that comes in here called DNA ligase. Ligase is an enzyme that's going to bond the DNA fragments together. They're not going to have like a smooth bonded backbone, so this ligase is going to bond them together. We're going to see ligase again in the next chapter, so don't forget about them. Single-stranded binding proteins, these are molecules that help keep DNA single-stranded. These are on the backbone of DNA. I usually call them like SSBPs just to make it shorter, but these are the proteins in the back here that will, again, just help keep the DNA from tangling and getting damaged. Now, I like to show this, again, this is the next layer deeper. The reason I like to show this is because there are more enzymes per se than just one DNA polymerase. You're going to get a couple that all have different functions. Like one's going to remove the primer, one's going to extend the chain, actually do the DNA polymerase. Another one, like there's a bunch of other stuff here that just is going on. So just understand there's another layer deeper when you go to college. Hey ligase, what have you been up to? Just making ends meet. Okay, we're going to go through these. Sorry. There's another video I'll show you during class. Another video, shall I do in class? And their meme. Okay, replication of DNA. DNA polymerase attaches at several locations along the linear DNA strand. So what happens is you're going to get these replication bubbles, and they extend out. And then when they fuse here, they're going to just make, again, you're just going to have the new DNA strand. But this helps us kind of get the DNA replicated quicker. So it's not just one location you're reading down. So it'd be like if you and like several other people read one book, but you each started at a different place, and you're going to get that book done a lot quicker than if you just read the book by yourself. Now, DNA polymerase is pretty accurate. However, about one out of 100,000 bases is an error. And so, what DNA polymerase can do is it can actually go backwards. I know I said that all the enzymes don't go backwards, but it's again, it still makes 5 to 3. It can recognize an error and replace it and fix the mismatch there. And that's good. We want DNA polymerase to be kind of foolproof. Now, not 100% because we do want mutations because that adds genetic diversity into a population, but it is pretty accurate. And again, even if you think about if you copied a book down word for word, you're going to make a mistake here or there. DNA polymerase is pretty accurate. You can see how there's a C in here that's not supposed to be here. Again, you want a T here. It can go back and actually replace that. I love that meme. I forget who made that. A couple years ago, a student made that. All right, we're not going to go any further than this. Again, so this is going to end the replication part of the central dogma. We're going to get to the actual central dogma tomorrow in class.